So thanks everyone. Today I'm going to talk about the consolidation of the long-term memory in diadigma semi-cousin, which is the insight and Jessa ever addressed a lot before. So I'm from UQ and as another PhD student from Pavlovian lab. So as we know that insect memory has already been identified as a case of a point. And surprisingly, like lots of mammals, like a human, we found that there's a lot of similarity of formation on the memory between human and insect. So before moving on to my project topic, please allow me briefly the diadigma semi-cousin again. It's a species, a parasitoid species of the Ichneumonidae. It deposits the eggs on the diamondback moths, larvae, aka is a DBM. So it's already been found predominant parasitoid in the brassica vegetable field, also is well established worldwide now. So as we know, that's lots of flying insect in this such a multi-trophic interaction. The herbivore-induced plant odor, plant volatile would be information for them to locate the right food sources, even avoiding some predators. So like herbivores and pollinators, and sometimes there's the microbes and carnivores like predators and parasitoid. However, are those HIPVs as a reliable information about the host and the food sources to those natural enemy? So here rests the profound question that's reliability to detectability problem is a main issue for now. So as far as we know, that parasitoid, they have a flexibility to locate the host, even differentiate between complex volatile blends sometimes, but it really depends species to species. An attractive HIPV emitted by the non-host infested plants and also have been found to have a strong potential to confuse them, such as a non-wording experiments like pathogen associative Q or non-host associative Q may result in slightly confusion to those parasitoid. So what is the connection between HIPV, between learning and even the memory consolidation? So here I'm going to show some important discoveries. As we know that previous study have shown innate preference can be overridden by the experiences. It means that even these naive parasitoid, they have innate preference for some specific odor, but after learning with the new plant odor, this preference can be changed. And the associative learning also consolidates the memory, and it has been classified into three main phases. The first one is short-term memory. Seriatically, usually when within 2 hours, and the mid-term memory usually rest up after 2 hours, but it will wane after 24 hours. So the long-term memory usually can sustain at least 72 hours, even longer at a certain level of memory. So more interestingly, lots of study have found that long-term memory normally formed after a repeat learning events, especially the spaced learning. As we can see in this figure, only the 10 times of spaced learning can that the Drosophila fruit fly keep a certain level of memory. So here I'm going to show you the experimental method in my chapter in my project. It's a classical conditioning, aka the Pavlovian conditioning. So before conditioning, there will be a conditioned stimulus, which is the DBN infested plant. So during conditioning, I have three different kind of the types of condition. The first one is single oviposition. The second one is the mass condition. It means that I give this my diadigma wasp to have 3 times of oviposition in a row. So what the difference is different between mass and the space learning, a space condition. Space condition is mean that there will be a short 10 minutes of the break between 3 times of oviposition. So after this, I will use the pairwise of olfactometer's assay to assess whether whether they keep the memory for the learned order. So let's jump into the results section. So here, as you can see the left panel, you can see there's a setup of olfactometer assay. And our groups ever found that the innate preference of diadigma is for the DBN Chinese cabbage. But after learning with the DBN infested common cabbage, this preference had been changed in 1 hour memory. So it raises a question, how is going to happen if we train them with a different learning module, and how long it will sustain. So the innate preference on the left and the learn order on the right, as we can see in this figure. So with the single oviposition trial, we can see the memory for the learn order only sustain until 12 hours. We think that between 12 hours to 24 hours, the memory went. Interestingly, if we retrain, experience the diadigma wasp, female wasp, to have a oviposition again, their memory for the learn order can be retrieved. However, they don't discriminate the learn and innate preferred cues in 48 hours and 96 memories. So in the mass oviposition on the left, you can see that it shows the similar results to the single oviposition trial. However, in the time-space oviposition, we found they last significantly longer time of the memory for the learn order, which is the 24 hours. However, they still don't discriminate the learn order and the innate one in 48 hours and 96 hours. And interestingly, we found there is a relatively high proportion of the non-respondent individuals here. So our best speculation is that they get confused and they can't differentiate such a subtle differences between the learn order and the innate preference, which is the DBN Chinese cabbage. So give us brief summary here. Time-space oviposition to help them to retain a longer memory. If you compare to the mass oviposition and the single oviposition trial. However, they don't discriminate the learn order and the innate preferred cues on 48 hours and 96 hours. And we also found there's a higher proportion of the non-respondents here. So I already told you that our best speculation is that they get confused here. So our results suggest the different results to the previous study, which is the similar study in Cotacea rubricula. They require also require the 3-space trial to form the long-term memory. And they don't the long-term memory didn't completely consolidate here. However, the level of memory retained didn't revert to the innate state. So here I'm gonna have a great thanks to all of my members in my lab. So thanks.
Yeah, thank you very much, Hark. That was a nice talk, and we have time for 1 or 2 quick questions if anyone has a question before we proceed. I will ask you one question.
Yep.
Oviposition is oviposition in this parasitoid governed by only olfactory cues or volatiles, or does it also use arrestants like short-range cues?
Short-range cues.
Yeah.
Oh yeah, I trained them with the short-range cues because I put them in a small cage.
Yeah.
And those DB and infested plant is put in the center of the cage. Yeah. So once I release the wasp, I let them to experience the whole process. Okay. So first is the first encountering the plant odor, obviously, right?
Yeah.
Then they will lingering in the cage for a while, then start to alight on the plant, yeah, and crawl to the deviant larvae, then deposit the eggs. They might attack a couple times, then deposit the eggs. Yeah.
All right.
Yeah.
All right. Thank you very much, Hark.